Good afternoon. I'm Xiaoyue from the Drone Lab at Carnegie Mellon University. My talk today is on proving domain architecture design using language models. So what are multi-domain protings? Multi-domain protings are mosaics of structural functional units called domains. And domains are sequence fragments that encode a distinct structure or function. And domains are found in otherwise unrelated protings, and they can fold independently in many different contexts. And so, for example, for the protein MPP6, it has 5 domains, and they appear here all as separate folds. And the domain architecture of a multi-domain protein is the entity sequence of its domain content. And so, for example, for the MPP6, the domain architecture is L27, PDZ, SH3, and GOOK. And the domain architecture representation is a useful representation, and it's being used by all these different kinds of studies. And here I'm showing you an example of a multi-domain family with variable domain architectures. This is the membrane-associated guanylate kinase, or the MAGOOK family. So domain architectures in this family have similar domain content, but with variation. And the domain architecture of a multi-domain protein is intimately related to its function. For example, these 4 domains in the Cas protein, they all bind to different things. And this makes the Cas protein function as a scaffolding protein that groups these proteins together. And the MAGOOKs that I just showed you are just one of many families with these characteristics. And here are 2 more. Here's the receptor tyrosine kinase family and the non-receptor tyrosine kinase family. And these are all examples of large multi-domain families that emerged at the beginning of Metazoa, and they function in signaling and cell-cell interactions. And within each family, the domain architectures all have similar domain content with variation on a theme. And so how did these families come into being? Our mental model of how multi-domain architectures evolve is through also you have through domain gain and loss. For example, you have this gene with this protein with this brown domain, and with a domain insertion, you can gain a pink domain. And then there's domain duplication, where you gain another pink domain. And then with gene duplication, you now have 2 copies. And you can lose a domain and then get another green domain inserted. And now we have this family of domain architectures encoded by genes that share common ancestry. And so in theory, these processes have the potential to generate any domain combinations over the course of evolution. But in fact, we only see we see fewer domain combinations than expected by chance. So for example, the number of distinct domain bigrams in the randomized genome exceeds the number of distinct genuine bigrams by a factor of almost 5. And for trigrams, the randomized trigrams is almost 8-fold greater than the number of genuine trigrams. And another striking example is that zinc finger and immunoglobulin are the most common primate domains, but they never co-occur in the same protein. But they co-occur 344 times on average in randomized domain architectures. And this suggests that there are stringent constraints acting on domain adjacency and co-occurrence. And so we want to understand these constraints. And so we, as a first step, we got a domain architecture simulator to mimic these constraints. And this gives us a handle of what the issue might be. And the simulator uses a sampling process guided by genuine bigram statistics. And I spoke last year at ISMB in this track about this project. And this year, I'm going to talk about modeling domain architecture space using natural language processing methods. And this framework can first help us interpret the results of the simulator. And it can also help us investigate relationship between domain architectures. And in general, this framework allows us to ask questions about sets of domain architectures in a unified setting. It supports exploratory analysis of the universe of domain architectures, comparison of multi-domain complements across taxonomic lineages, and testing hypotheses about domain architecture evolution. And to do this, I'm going to use this metaphor, where in natural language, documents and sentences are strings of words, and domain architectures, they're actually strings of domains. And so NLP seeks word embeddings such that words with similar meanings are close in the word embedding space. And documents with similar content or topic or sentiment are close in the document embedding space. And with this metaphor, I want to embed domains and domain architectures in high-dimensional space. And I want domain architectures that perform similar functions to have small distances to each other in the space. And to do this, I'm going to do this in 3 stages. I'm going to tell you about the 3 steps, and I'm going to show you 2 examples of how I can use this framework. So in the first stage, we construct domain embeddings. And each domain D is represented as an m-dimensional vector E of D. And the dimension m depends on the specific information retrieval or natural language processing technique that are used to construct the domain embedding. And in this work, I'm going to show you 2 examples, TF-IDF and Word2Vec. And a few other people also did this with Word2Vec, and they did domain embeddings. And in my second stage, I also do domain architecture embeddings. I construct domain architecture embeddings from the m-dimensional domain embedding and represent domain architecture A as a vector V of A. And this can be calculated from the embeddings of the constituent domains. And specifically in this work, I'll take average of constituent domain vectors. So for example, the embedding of domain architecture A sub 1 is just sum of 2 oranges and 1 purple domain plus 1 green domain plus 1 brown domain divided by the length, which is 5. And there's comparative analysis once you have the embeddings. And I'm going to show you 2 examples. Both examples are based on the set of unique domain architectures in the human proteome. And I obtained the domain architectures from the superfamily database, which is a collection of domain models based on structures in SCOP. So the first application is comparing sets of domain architectures. For example, we can ask, how does the multi-domain proteome differ across species? I first developed this framework because I wanted to compare simulated domain architectures with real domain architectures to assess how well the simulator recapitulates properties of genuine domain architectures. So, I first asked whether the set of simulated domain architectures recapitulate properties of genuine domain architectures. I used a set of genuine human domain architectures as the training corpus for my domain embedding. And based on that, I construct domain architecture embeddings for both the set of genuine domain architectures and simulated domain architectures. And now the 2 sets of architectures are represented as points in the domain architecture space. And to assess how similar the 2 sets are, I ask what is the smallest perturbation required to superimpose the 2 sets of points. And this is called the Earth mover's distance. And here you see 2 embeddings, one generated with Word2Vec and one with TF-IDF, and they're shown as 2D representation obtained with t-SNE. And red points correspond to genuine domain architectures, and blue points correspond to simulated domain architectures. And you can see that the red and blue points, they are pretty close in this domain architecture space, but how close are they? So we need a baseline comparison. So for a baseline comparison, I included a set of randomized domain architectures. And once I've put the randomized domain architectures, which are yellow points, into this space, I see that in general, genuine and simulated domain architectures, the red and blue points, are in close proximity relative to the yellow randomized domain architectures. And so now I want to quantify this relationship. So I use the Earth mover's distance to confirm the observation. Between the red genuine set and the blue simulated set, the distance is much smaller than between genuine and randomized, or between simulated and randomized. And this is the case for both embeddings. And so this experiment shows that our simulator, the Markov, was able to recapitulate the properties of genuine domain architectures better than expected by chance. And so that's my first example. And in my second example, I'm going to show you how a visual representation of an embedding of human domain architectures can be used to explore relationships in multi-domain families. So people like to group proteins into families. But what are protein families? Protein families are defined in various ways. Some people think of them as homologous sequences that share common ancestry. And some people think of them as sequences that share common functions, but often homologous families also share common functions. But multi-domain proteins, they push the boundaries of these definitions because of their modular nature. Some part of the sequence might share common ancestry and some others don't. And so my framework can help us explore this boundary. As a test case, let's look at these three families that I showed you earlier. And I put them in the context of the human domain architecture embedding. I'm going to project them into the domain architecture space. And I'm going to see where they are and what are their relationships. So this is a 3D representation, again, generated with t-SNE. And I have highlighted the McGucks in purple, the non-receptor tyrosine kinases in blue, and the receptor tyrosine kinases in pink. And the first thing that you notice is that these three families, with a few exceptions, they form well-defined clusters within the embedding. And to understand the relationships more, we can take a look at their domain architectures. So these are the two families, right? The RTKs and NRTKs. And the receptors, they all have this transmembrane region where they're anchored to the cell membrane. But the non-receptor tyrosine kinases, they don't have the transmembrane region. Also, this is a marker that can separate the two families. But actually, transmembrane regions, they are not structural domains, and so they're not included in the domain architectures that we obtained from superfamily for this study. And so even though transmembrane regions are not included in our data sets, the embedding is still able to separate the two families because each family is characterized by distinct auxiliary domains. And a likely reason for the distinct N-terminal auxiliary domain is that the N-terminal region for RTKs are extracellular, and the N-terminal region for NRTKs are intracellular. And our next observation is that in the NRTK family, in the RTK family, there are a few domain architectures that are outside the main cloud. And let's look at MET for an example and see what we can learn from it. So with the exception of the shared kinase domain, all other domains in MET are unique within the RTKs. And in particular, the MET domain architecture includes a SEMA domain and a plexing domain. And here you see it again. I have highlighted those domains in green, blue, and pink. And these domains are unique within the RTK family, but there are actually two other families that have the SEMA and plexing domains, and they are the semaphorin family and plexing family. And we can use the embedding to ask, what is the relationship between MET and these two families? And if we zoom in to the neighborhood of MET, does it cluster with semaphorin and plexin? Well, yes, indeed, MET is tightly clustered with plexins, and they are very close to the semas. And this raises questions about the functional relationship between MET and semaphorin and plexin. Well, in fact, they are functionally related. MET and plexin B have identical domain architectures except for the intracellular domains. MET is involved in a number of different pathways. For its canonical pathway, it dimerizes with itself. And in one of the non-canonical pathways, it actually binds the plexin B and triggers downstream signaling. And the proximity of MET to the plexins and semaphorins also raises questions about evolutionary relationship between MET and plexins and semaphorins. And so a recent comparative study by Alves et al. Suggests that MET is the result of a kinase domain being inserted into an ancestral plexin. And so possibly this MET protein evolved differently from other RTKs. And in fact, perhaps we should consider MET to be a member of the plexins rather than one of the RTKs. And it is surprising to me how we don't actually use sequence. We don't use amino acid sequence, but we are able to spot this relationship between MET, the other RTKs, and plexins and semaphorins. And so in summary for this example, there are three families, RTKs, NRTKs, and MAGUCs. We see that they form three distinct clusters, and domain content distinguishes the receptors and non-receptors. And the outliers in the domain architecture space, they actually correspond to biological outliers. And by now, I hope I have convinced you that this framework is useful in exploring the relationships between domain architectures and generating hypotheses for us to explore. And so for conclusions of this work, domain architecture embedding framework supports visual and quantitative analysis of domain architecture data. It has many applications, including exploratory analysis of the universe of domain architectures, comparison of multi-domain complements across taxonomic lineages, and testing hypotheses about domain architecture evolution. And our framework provides a nuanced representation of domain architectures, because the domain architecture vector representations is based on a domain embedding. And the relationship between the domain architectures not only depends on shared or not shared domains, but the contribution of domains that are not shared depends on the distance between them in the domain embedding. And I would like to thank my lab, my advisor, Danny Durant, and these people who kindly gave advice and suggestions. And I would like to thank my funding resource. And thank you all for listening.
I think I can take one or two questions or comments from the audience. So if you have any, please raise your hand. Sure.
So I guess you're not using any sequence information, but could you see a way that you could combine the domain mapping and also use sequence differences between domains and get a better resolution somehow, or the evolution history?
So the question was, this framework doesn't use amino acid sequences, and is there a way we can combine sequence information and domain content information? I think you could, I guess, refine the distances that you obtained with sequence similarities. I think that is one way. And to be honest, I didn't sleep at all last night, so I can't think of anything else. But if I do, I'll let you know. I think that is one interesting question. I don't know if anyone else in the audience has an opinion.
Does it give you the same relationship? You compare sequence distances and...
No, sometimes it doesn't. For example, for the RTKs and GOKs, usually these multi-domain families, they only share a subset of their domains. So their trees are actually, if you want to build trees, you anchor one domain, you build trees with that domain. And actually, this relationship that you obtain from this tree is not always the same when you consider domain architectures and domain compositions.
Okay, thank you, Xiaoyue.